If I were to ask your mom what you're working on, what do you think she would say?
God. I actually don't know if she knows what I'm working on right now.
What would she say?
He's working with mosquitoes and trying to understand the brain. That's probably the extent she'd say.
Dr. Kevin Nunez is a researcher at NYU who studies fruit flies, not mosquitoes. As an undergraduate, he researched the effect of alcohol on brain health using human samples. As a PhD, he kept exploring alcohol in the brain, but switched from human samples to fruit flies.
They understood my undergrad work a little bit more. I started getting further into the flies, like, wait, you're doing this all in mosquitoes, like little flies? And in this case, they were thinking like mosquitoes themselves. I'm like, no, not doing in mosquitoes. I don't actually remember the name of how to say fruit flies in Spanish, but we had like little nicknames for them, like the things that go around the fruit when you leave them out. I started losing them a little bit then. They were just a little confused.
It's not just Kevin's parents who get tripped up, but politicians too. Here's Senator Rand Paul talking about work into how biological sex influences aging.
They spent $1 million trying to determine whether male fruit flies like younger female fruit flies. I think we could have polled the audience and saved $1 million.
And former Alaska governor and vice presidential candidate Sarah Palin talking about research into the genes that cause autism.
Sometimes these dollars, they go to projects having little or nothing to do with the public good. Things like fruit fly research in Paris, France. I kid you not.
You can find jokes about research in fruit flies everywhere, especially if you combine that with drugs, like researchers did when they looked into genes that might make us more susceptible to cocaine addiction.
Wait, but why did these scientists do that?
Because they were also doing cocaine.
They were like, you know what'd be really cool? All right, you know what'd be really cool? How about?
But there is a certain segment of the population that loves fruit flies.
Flies are cute. If you ever look at a zoomed in picture of a fly head, they're super sweet. They got cute little antennae with little feather on them. And when they clean themselves, they just look adorable. So here's some fly propaganda. They're adorable. Go Google them. I love them.
Fruit fly researchers revere fruit flies, and not just because they're cute. The first thing a researcher will bring up if you show any doubt about the importance of fruit fly research is
the Nobel Prize was for the work in fruit flies.
I think there are 6 or 7 Nobels now given out for work that was primarily from the fly model.
6 Nobel Prizes awarded for work done in fruit flies. That's included research into the basics of how traits are passed down in families, how the body fights off infections, and the internal clock that controls our sleep and wake cycles. But why fruit flies? I'll leave that to Folke Henschen, who gave this speech when awarding the first fruit fly-related Nobel Prize to Thomas Morgan back in 1934. And by that I mean my friend Josh reading the speech in an old-timey voice.
Another cause for Morgan's success is no doubt to be found in the ingenious choice of object for his experiments. From the beginning, Morgan chose the so-called banana fly, Drosophila melanogaster, which has proved superior to all other genetic objects known so far. This animal can easily be kept alive in laboratories, can well endure the experiments that must be made. It propagates all the year round without intervals. Thus, a new generation can be had about every 12th day, or at least 30 generations a year. The female lays about 1,000 eggs. Males and females can easily be distinguished from each other, and the number of chromosomes in this animal is only 4. This fortunate choice made it possible to Morgan to overtake other prominent genetical scientists who'd begun earlier but employed plants or less suitable animals as experimental objects.
So, lots of reasons to love flies. You can make tons of them super fast, which is convenient if you're trying to understand how genes are passed on between generations. And what scientists have first discovered in flies has often been found to be true in humans. Nowadays, fruit flies are very popular among neuroscientists. They have brains, but they're much smaller and simpler than ours. giving scientists a more manageable way to study the fundamentals of brain circuits.
If you come at it from a neuroscience perspective, it really opens up the kinds of questions that you can ask. You can really dig down into how precisely certain circuits work in the brain, not just sort of broad circuits of many neurons, but like little loops of like 3 neurons that talk to each other in some sort of feedback loop or something like that. And you can really probe how the brain works on a more microscopic level, I would say. And even more than that, how pared down can you get and still do really cool things? Because the other thing that's really cool about flies is that they are insects, and insects are an ancient form of life. And I love thinking about how much time evolution has had to really trim down and make as efficient as possible the fly brain versus the human brain, which has greatly expanded in size and neuron number from, the great ape primates that we evolved from. Now, obviously, humans have way more behavioral niches. I'm not expecting my flies to write a symphony or something like this, or even write anything at all. I think of the fly brain as like the Lamborghini. It's really sleek and it's really good at what it does, which is get a fly around so that it can find food and mates. And that's really all it has to do. And it does an excellent job of that.
Wow, Lamborghinis, Nobel Prizes, all sounds pretty great, right? But this podcast is called Frivolous Science, not for nothing. We are not talking about groundbreaking disease-curing research. Nope. Today, we are talking about the science behind food comas. Yes, that drowsy feeling after a nice big meal. Does it happen in fruit flies? Why would anyone bother studying it? Don't nod off yet. We're about to get into it. In Frivolous Science, we talk about whether research should get funded or not, but at this moment, a lot of it isn't. Yeah, how's your lab doing?
So my lab is always on the brink of death. If we're not fully funded, the lab closes down. And so anytime one of our ongoing grants is near the end, about to expire, we're always nervous about the next one because we cannot really be in between grants. We have to have the next one to keep the lab going, or the lab shuts down. And usually I'm not so nervous. Usually something picks up that saves us and keeps us going for another few years. But this time the market or the overall environment is much more challenging. With the defunding of NIH and other biomedical research institutions, it's just much more difficult overall.
This is Dr. William Jha, a professor at Scripps Research Institute, Florida campus.
Yes, except our institute has actually changed. And so the institute name is now a mouthful, but it is, hold on a second, let me get this right. We are the Herbert Wertheim UF Scripps Institute for Biomedical Innovation and Technology. But I usually call us UF Scripps for short.
Okay, UF Scripps.
Yeah.
Okay. Before we got to his study, I wanted to know more about his motivation for studying flies in the first place. So I'd love to hear, before we get into the details of the food coma study, I'd love to hear your motivation for working in flies, because flies are pretty distant from humans, right? We already get a lot of people talking about, why do we study mice? Why not study humans? And flies feel even further away. So can you comment on that? Why would you want to use flies as a model for biological processes?
Sure. Each of the popular model organisms we use in science have different advantages and disadvantages. With flies, there are some very clear advantages for why we use them. Powerful genetic tools allow us to manipulate genes, insert disease genes, make mutations, and test them in a living model. They are cheap to maintain. They're easy to grow up hundreds or thousands of animals to use for our experiments, enabling high throughput studies. For me personally, my first projects in the fly were on aging. And so we were looking at ways of identifying or understanding genetic mechanisms and other environmental manipulations like diet or nutrition that affect aging, that extend longevity or extend fly lifespan. And for aging research in particular, using mammalian models is very difficult because of the long lifespans involved, right?
Sure, yeah.
Mice live several years, so if you want to test the idea that this diet or drug extends mouse lifespan, it's a multi-year experiment. Whereas in flies, they live about 1 to 2 months, 30 to 60 days. And so it's much easier to test hypotheses or test diets or drugs or whatever to see if you've extended fly life, and then to do those genetic screens to identify the mechanisms that are involved for that longevity phenotype. And then you hope that what you learn provides some models or ideas that you can then push forward into either mammalian models or, even preclinical or clinical studies.
Yeah, that's so interesting. I mean, it makes sense from a practical standpoint. If you're working at the timeline of a scientist conducting a study, if you want to get answers, that you would try and pick an organism with a shorter lifespan.
Right. And that's specific for aging research. But in our other work that segues into our neurobiology or behavioral work, we've always been really interested in trying to understand how a brain works. Mm-hmm. But a brain, especially a human brain or a mammalian brain, is very complex. And so one of the ways you try to figure out or dissect how our brain works is to use a simpler model system. And the fly is a fantastic balance between simple and complex. The fly does have a brain. It's much smaller than ours, obviously. It has much fewer neurons and connections. Yet this simple brain enables a bunch of reasonably complicated behaviors. They can make choices. They eat. They have to make feeding decisions. They sleep. They have circadian rhythms. They fight. They show aggressive behavior. They have social interactions. And so this rich repertoire of behaviors in a simpler brain allows us to try to dissect how a simple brain regulates these behaviors. And hopefully that teaches us some of the strategies that a more complex brain will use to regulate similar behaviors.
But we're not just here to talk about how great flies are. You've already heard about all of those Nobel Prizes. Let's get to the part about food comas, or as Dr. Jacques calls it, postprandial sleep, which literally just means post-meal sleep. So then getting to the paper that first drew me to your work, where you identified this change in sleep behavior of flies after they ate.
Right.
I'm really curious about how you even got to look at that.
Well, I guess that's a good story. It was not born out of any hypothesis-driven research. And I know that's having a hypothesis and then designing experiments to test that hypothesis is a very important part of science. But a lot of the stuff we do in the lab is exploratory and, fun. And so that project came about because we were interested in the regulation of feeding behavior, understanding how animals decide when and what to eat and how much, and so forth. And to do those kinds of experiments in the fly, we had to develop new tools or new techniques to quantify feeding behavior. Basically, we use 3D printers to make arrays of enclosures to keep individual flies in. And we put a little food in there and flies can eat the food. And we have video cameras tracking the flies in each of these chambers. Each fly is individually housed in a vertically oriented chamber that doesn't have too much depth, but there's enough depth for them to walk around. And the way we measure feeding behavior is by putting liquid food. So these are food solutions, usually yeast and sugar water, that we put into a glass microcapillary. So it's basically a tiny glass straw. We have a video camera or webcam just recording the straws and watching the food move, and we can calculate the amount each fly ate in each chamber.
And then so you're using the capillaries, watching that to track how much they ate.
Correct.
And at the same time, you're tracking their movement, right?
Right. And from locomotor activity, at least in flies, you can also derive sleep. It was shown previously by Paul Shaw and other fly sleep researchers that periods of immobility where the fly hasn't moved for 5 minutes or longer are highly correlated with sleep. That it's highly likely that the fly is sleeping if it hasn't moved for 5 minutes. And so we had all of these behavioral metrics from a single recording, from a single video. And we just wanted to know what is the coolest thing we can analyze from this giant, pot of data. And maybe more specifically, we had this tool that no one else had. And so another question I gave to the grad student, Keith Murphy, who pioneered this work, was what is something that our device can measure that no one else can, at that time. And so we came up with the idea of trying to do this feeding-sleep correlation. At that time, there was a lot of evidence that diet and nutrition and feeding behavior is associated with sleep, but at least in flies, it was very challenging, if not impossible at the time, for researchers to capture feeding and sleep data from the same individual fly.
Okay. But this setup was perfectly built to be able to do that.
Right. So we have all this feeding and food intake data and sleep and locomotor activity and all these metrics for individual flies. And we asked, what is the coolest thing we can analyze out of it? And so postprandial sleep or food coma was an easy idea that popped into our heads. We just said, do flies show food coma? Do they sleep after eating? And it wasn't entirely clear how we analyze for that. If you can imagine, flies are feeding from the straw, they suck from the straw, they run away, they come back, they suck from the straw again. How do you turn all these feeding events into something we could call a meal? And do flies eat meals, or are they constantly grazing? Are there discrete enough feeding events that we can try to see if there is some sort of sleep associated with each of these feeding events? And so we eventually did a bunch of things to show that, yes, flies do eat what we are calling discrete meals. They eat 5 to 12 times a day, either bigger meals or smaller snacks, but they are discrete feeding events. And so by recording 60 flies over several days, you get hundreds or thousands of meal data points. And we had no real idea of how to analyze for postprandial sleep. And so I feel like what we did was very naive, but it seemed reasonable. We basically took these 1,000 meals that we had, and we lined them all up at t = 0. And we simply asked, what does sleep look like before and after each of these meals?
Okay, so you just, for all of the flies all together, every time it ate, you just counted that as time = 0. And then you measured 20 minutes before that and 20 minutes after that, right?
Right, roughly 20 to 30 minutes. Another way to say is we basically took all the feeding events, the meals, and we lined them up together at the same time. It doesn't matter if they ate in the morning or at night or in the middle of the day. We just lined up all those 1,000 or 2,000 meals, lined them all up at the same time, and ask, what does the average sleep look like for the 20 or 30 minutes before that meal and the 20 to 30 minutes after that meal? Does that make sense?
Yeah.
Okay. So at t - 30 minutes to the meal, there's some average baseline of sleep. And as you reach the countdown to t = 0, that sleep plummets to almost nothing. And that makes sense to us because flies cannot sleep and eat at the same time, right?
Yeah, we're not sleepwalking, eating.
Right, so sleep plummets. And then immediately after t = 0, after that meal, you see that average sleep start to come back up, start to rebound.
Yeah.
And so it looks higher on the post-meal side than the pre-meal side. And we did a couple different things to analyze that further. We summed up all that sleep post-meal and compared it to the same amount of time of sleep summed up pre-meal. And we showed that the post-meal sleep was more than the pre-meal sleep. So that's one way to do it. Another way you can do it is just to plot it, and you can see it temporally, that as you get away from the meal, that sleep rebound from 0 to 15 or 20 minutes after the meal is higher than the pre-meal sleep. Does that make sense?
Yeah, for sure. So then on average, these flies tend to be more likely to be sleeping after a meal than before a meal.
Right.
That's right. How much variability did you see from fly to fly?
A lot.
Yeah.
A lot. And from meal to meal. And so that's one of the things that was kind of exciting by this work when we dug into the literature, because we all anecdotally feel like food coma is a real thing, right? Especially after a big meal or a buffet, I get sleepy. How have people not studied this for years and years? But it turns out anytime, there are only a few papers on this, but anytime people try to study food coma or postprandial sleep in humans, they get a very variable result. Sometimes they see the effect, sometimes they don't. Sometimes the subjects get sleepy after eating, and sometimes they don't. So it's just a tough thing to capture or record. And if you believe our fly data, right, if you believe that what we're studying is something similar, it might explain why it's been so hard. So for any individual meal, after that meal, the effect on sleep is a very broad range of phenotypes, ranging from slightly arousing, right? So that the fly didn't sleep more, it was slightly aroused afterwards, all the way to deeply sleep inducing. And so the only reason we see this average increase in postprandial sleep is because we have 1,000 to 2,000 meals as data points to work with. And that's something that's very tough to do in mammalian models, to get that much data.
Yeah, so this is one of the really big advantages of flies in general, where you can analyze so many, and also your specific system,
Right.
where you're assaying 20 or so at a time for a long time in an automated fashion. So there's not someone who's checking a box every time a fly is eating. So
Right.
it's something that you can scale up.
Right, exactly. And this initial analysis, this idea of increased sleep after eating these meals, wasn't enough to convince us that we were seeing real postprandial sleep either. There was a number of experiments to follow that up that finally convinced us that what we were seeing was real sleep, and it was really happening after meals.
So this is one thing that you mentioned, was that there's previous data showing that inactivity means that a fly is asleep. Inactivity for 5 minutes is highly correlated with sleep.
Right.
But then you went on to check, is that really the case in our data?
Right. So that's one of the things we did, was to try to do another test of whether they're sleeping or not. The not moving for 5 minutes is a pretty good measure, and it's really easy to make. But if we're trying to come up with this brand new phenotype, this postprandial sleep phenotype, it would be good to double check that that was real. And so we did that by quantifying something called arousal threshold. If you see someone resting or not moving, how do you know that they're really sleeping or just not moving because they're resting, right? And the way you test that is because if they're really sleeping, they have a higher arousal threshold, meaning when you poke them, it takes a harder poke to get them to move if they're sleeping. Whereas if they're just resting, it takes a lighter poke. And so we did an arousal threshold test with the flies after meals by basically gluing cell phone vibrator coins to the back of the chambers and increasing the amount of force of those vibrations, the G-force of those vibrations after meals, and showing that after meals, the arousal threshold did indeed increase, suggesting that it's really sleep.
And did you actually get those from cell phones?
Oh, you can buy them from some electronic shops or AliExpress or something like that.
So you just gently vibrated the flies at first and then increased it, and you saw which level of vibration led to them
That's exactly right.
moving.
That's exactly right. You start at a low level vibration, you increase the force of that vibration. I forget what the program was, every 5 seconds or 10 or whatever seconds. And then the level at which they start moving is the threshold that you score for that fly. So we saw that there was an increase in arousal threshold after meals, suggesting that increase in post-meal sleepiness is really sleep and not just post-meal restfulness, or maybe rest is the wrong word too. It wasn't just post-meal immobility, right? You can imagine if you eat a lot, you just don't feel like moving, but they might not be sleeping.
Yeah, no, that makes sense. So you're making this distinction between stillness and sleep.
Right, exactly.
Cool. Yeah. So you found out this phenomenon. You're able to confirm rigorously that there's this postprandial sleep, post-meal sleep in flies. And then the rest of your paper is really looking more deeply into the mechanisms regulating that phenomenon, right?
Yes. Yeah.
So one of the things you looked at was how much they ate, right?
Yeah, just another way to break down that data. When I first described how we analyzed the data set, we took those 1,000 or 2,000 meals and we lined them all up. But by feeding the flies different diets that have more protein or salt or sugar, we can take those 1,000 meals and bin them in different ways and analyze the data again. So the first thing we did was to take those meals and separate them into big meals, medium-sized meals, and small meals. And you can see that the bigger the meal, the more postprandial sleep they show. And at least anecdotally, that makes sense, right? It's when we eat the big meals at Thanksgiving or at the buffet that we get our belly's really distended and I think anyway, that we feel the most sleepy after eating.
Yeah.
So the meal volume thing made a lot of sense, or we liked it anyway. But the other way we analyzed the data was to take, again, those 2,000 meals, and then we first threw away the big meals because we already know the big meals, meal volume drives sleep. So we just take the small meals, right? Now from those small meals, we can bin them separately into meals that had a lot of protein, a medium amount of protein, or a small amount of protein. And we can do the same bins for carbohydrates or sugars, high sugar, low sugar. And we can also do that for salt. So high salt, medium salt, low salt ingestion with these small meals. And that analysis revealed specific meal components or ingredients that also drive this sleepiness after eating. And I think we were a little surprised because the drivers of the postprandial sleep turn out to be protein and salt, but not sugar.
Yeah, I wonder what you made of that because it sounded like there was already data showing that sugar modifies sleep in flies in some way, right?
Yes. But if I remember, those are usually, if you change the diet completely and leave them on that for a while, how does sleep homeostasis or sleep balance change? So this is sort of more of a long-term effect. Whereas postprandial sleep is really an acute or short-term effect that says, when I eat that meal right there, if there's a lot of sugar in that meal or very little sugar or a lot of protein in that meal or little protein, what happens in the next 15 to 25 minutes afterwards to how sleepy I feel? So it is a different question when we're looking at it from this post-meal perspective. And you're right, we did find it kind of surprising because we thought, again, all of these ideas come anecdotally, right? But we were thinking kids and Halloween and sugar, eating too much candy and the sugar crash, so that these flies would also crash and get really sleepy. But they didn't. And in hindsight, maybe that makes sense. I think we had read or seen somewhere that this sugar crash temporally comes a bit later. Like after you ingest sugar, there's some brief hyperactivity. And if there is a real crash, if that's real, it comes a bit later. So it's just the timing may be different for that than what we were seeing immediately after meals.
Right. Did you ever go back and look at that to see, because you already have that data, but you only really looked at those 20 to 30 minutes before and after?
Right. Yes, but I don't think you can get anything out of that. It's really difficult because keep in mind, we're taking these 1,000 meals from all different times of day, right? Morning, night, and whatever, and lining them up at the same time and looking at the sleep before and after. If you start looking farther away from the meal, like 1 hour, 2 hour, 3 hours, the time of day that meal occurred really starts to matter because your circadian sleep, the regular time that you sleep, starts to make a difference. Flies, like us, sleep more at night than during the day, and that regular sleep pattern starts to make it challenging to see any longer term effects from meals.
Right. So whenever you're doing these additional manipulations, you're trying to make this comparison of some baseline to, let me just change this one small thing, which is part of the benefit of working with these fly models. But no matter what, you still have so many other aspects. Food isn't the only thing that's affecting sleep. Like you said, it's circadian rhythm, individual variability, and probably some other factors.
Right. But we could do something, we didn't do this, but we could do something like you suggest. We could go back and take only meals that the flies ate between 11 a.m. and 12 noon. And then ask, what does sleep look like between 2 and 6 p.m., so a few hours later, to look at a long-distance effect. And that would sort of isolate the circadian component, right? Try to narrow it down and only select meals from a certain time of day, and then look at sleep only a few hours later at another very specific time of day, not during the night when they're normally sleeping. And maybe that would show an effect, a longer term effect of sugar in the meal. But we never did that type of analysis.
So what did you make then? So there's the surprise about sugar not having an effect in the time that you're looking at. But what do you think proteins are doing then? And you also saw an effect with salt,
right? Sodium.
So what do you think? Sodium, yes. So what do you think both of those are doing that lead to this increase in sleep?
Well, with protein, we thought it was cute because the urban legend has always been that tryptophan from Thanksgiving turkey causes that effect. So protein seemed okay, seemed reasonable. Salt was a little bit of surprise, but we do put a lot of salt in our foods. At the time, we really didn't know. The data was what it was, and then we went on to try to identify neuronal or genetic mechanisms regulating the behavior. But we now have unpublished data that points to some possibilities. And so after that work, one of our unpublished pieces of data was to try to figure out what the purpose of postprandial sleep was physiologically. If you believe that we're studying post-meal sleepiness and that this is a conserved behavior that you see in mammals, then it must be something important, right? Something evolutionarily conserved across species should be important and do something.
And why? Why does it have to be important?
Well, I think the idea is that if it doesn't do anything, it would be lost, right? If it's a behavior or a gene, this works at many levels, right? Physiology, metabolism, genetics, behavior. If it's something that is not useful to the organism, it will be lost in future generations, especially something that seems like it's so costly to maintain, right? Sleep is sleep is necessary. That's why animals do it. But sleep is, especially in the wild, for wild animals, is typically a pretty dangerous thing to do. You're immobile. You're less sensitive to environment, right? Your arousal threshold is higher. You're more prone to predation. So it's potentially a very costly thing to do. So if postprandial sleep is maintained in animals, you'd like to think it serves some important purpose. And so one of the hypotheses, one of the ideas of what that purpose might be was something to do with digestion. That was the most obvious, right? You eat a big meal, you get sleepy, we thought it must help you digest or do some digestion. And so it wasn't easy to figure out how we would test that idea, but we developed a pretty cool experimental approach that's really hard to do, but we basically fed flies single meals that were spiked with radio-labeled sugar or protein, okay? And then we would feed them these single meals and then ask several hours later how efficiently they absorbed that sugar or protein.
Okay, how do you measure that?
Basically by seeing how much of that radio label, that radioactive tracer, accumulated in the fly body. If you see a lot of radioactive accumulation in the fly body, it means it effectively or efficiently absorbed that sugar or protein.
Versus releasing it.
Right. And we also had to collect the crap, right? The excrement, the excreta. And compare that. And if you see a lot of it in the excreta, then they excreted most of it and they did not efficiently digest it or absorb it. It's a real tedious experiment to do, working with individual flies, with individual meals, but the data is very quantitative and very, very cool to look at. And so in the control experiments, the first experiments, we showed that no matter how much sugar, sucrose, is in the fly meal, ranging from, I forget what it was, 1 to 20 micrograms of sugar in that meal, the flies absorb all of that sugar like 98, 99% plus. So flies are very good at digesting, processing, and absorbing sugar.
Okay.
With protein, however, ranging from a similar range in micrograms of protein per meal, you get this decrease in the percent efficiency of absorption. And so with very, very low protein ingestion, with low protein amounts in the meal, they absorb most of it, 95, 98% plus. But as the amount of protein in that meal increases, that percent efficiency of absorption goes down to 85 and then 75, I think down to, at the highest level in the meal, it was 60 or 70%.
Interesting.
So flies are very good at absorbing and digesting sugar, not so efficient at processing and digesting protein. So we were super excited by this because this matches the idea that, postprandial sleep must be important for helping with that digestive process, right?
Yeah, because if it was already very highly efficient, then you wouldn't need some additional mechanism to help with processing.
Right. And so we repeated that experiment with the protein absorption radio tracer, but this time after feeding the meal, we took half of the flies and we put them on a vortexer and basically shook them for a few hours.
Oh my gosh.
Not the best experiment, but they couldn't sleep, right, for those few hours.
Yeah.
And the other half of the flies, we just let them sleep after eating as they would normally do.
It's a gentle vortexer, right?
Yeah,
They're fine.
Yeah, but I mean, we could see that they were moving around, so they weren't sleeping.
Okay, yeah.
And so when we did this, and then we asked what was the percent efficiency, we were surprised to see that when you don't let them sleep, protein absorption efficiency very clearly went up. All right.
When you don't let them sleep, their efficiency goes up.
Right. They absorbed more protein. They were better absorbing protein.
Oh, that's surprising.
So that's true. That would suggest that the normal purpose of postprandial sleep is to actually reduce the amount of protein that's digested and absorbed.
Huh.
So then we had to go back and think, could this be right? Why would this be right?
Yeah.
And we came up with two things. So first, turns out that flies are actually, compared to mammals, pretty bad at getting rid of nitrogenous waste. Proteins are the major, nitrogen source from food. And because of that, high amounts of protein are toxic or detrimental to fly lifespan. And so is salt or sodium. Flies, being so small, are very sensitive to dehydration stress. And so salt or high amounts of sodium are also very toxic for them. And so both of these compounds, sodium and protein, induce this postprandial sleep effect. They're both bad for the fly. So maybe postprandial sleep is indeed, the purpose of it is to get rid of these components or not absorb as much of it when you eat too much of it in a big meal.
Interesting. And this fits in with the behavior of what they want to eat, right? What they go for. Do they usually go for foods that are more sugary rather than proteinaceous or salty?
That's hard to answer. So first, depends on sex. So the males and females have different protein carbohydrate targets. But then at a second level, when you let the flies choose between how much protein and carb they eat, they do generally choose, depending on sex, but they do generally choose this specific ratio of carb to protein. So it's hard for me to say whether that's a high amount of carb or a high amount of protein.
Okay.
Like you're asking me whether the flies usually go for carb or protein, and it's neither. They go after a balance. So if you let them choose, they go after a target. I can't really tell you if that target feels high protein or high sucrose, unless I'm comparing it to something.
Sure. Okay. No, that makes sense. I guess what you're telling me is your theory about post-meal sleep, and it's decreasing the efficiency of protein absorption. So I was getting a little speculative about other things that a fly might do to help it have less of that waste to begin with by consuming less of it when it's controlling its own diet.
Yes, it does regulate its feeding to choose the appropriate level of carbohydrate and protein.
Yeah.
But it could be that its major source of protein in the wild will be live yeasts, fungus, and bacteria growing on rotting fruits. And so it may not be able to say, I'm only going to have a little bit of protein. It starts eating these yeasts on top of the rotting fruits. And if that's a highly proteinaceous meal, it may balance that out by sleeping more after that meal and excreting more.
Yeah, that makes a lot of sense. Dr. Jha also discussed some human data that might support the idea that post-meal sleep is involved in regulating how nutrients are processed by the body.
The only other thing we have that made me feel comfortable with this result, even though we haven't published it yet, is there's one human study from, I want to say it's like 30 years ago now, where they took college students and they gave them a free meal, and then they let some of them sleep and some of them not, right? Some of them they didn't let sleep, and they measured diet-induced thermogenesis, which is a measure of how many calories you were able to process from that meal. And the result they saw was in the same direction as what we saw with our sleep manipulating experiments in the fly. There was more diet-induced thermogenesis when you don't let the people sleep after eating, suggesting they were able to extract, more calories from the food, not less.
Yeah.
So that was kind of exciting when we found that one paper, because it was consistent with what we, in the same direction that we saw, and might suggest again that the phenotype we're studying is something conserved across species, and that gives us more motivation to dissect it further.
Okay, interesting. So if you were, I guess you're working on flies, but there's this idea of it connecting to humans. If you were to design a human experiment that kind of extended what you were doing, do you have any ideas of what you would love to see?
It's a great question. I guess I'd love to see some of the stuff that was already done in humans repeated with bigger numbers. It's a little bit, I don't want to say disappointing, but disheartening, because the old literature, it was hard to see a consistent effect. And one of the leaders in that field, or the guy who was doing the most work, published the most papers on this, it was his papers that would sometimes show a postprandial sleep effect in his human experiments and sometimes not. And so he wrote a opinion paper, a perspective paper years later, lamenting, I guess, why is it so hard to measure this phenotype in humans that we all sort of anecdotally feel or feel like it is real and exists. And he just said, well, in the lab environment, it's just tough, right? First, sleep experiments in humans is tough. You have to do it in a sleep lab, and people just don't like sleeping when they're being watched. It's already you've already really perturbed them by putting them into this environment to do the experiment. And then circadian variables, just a lot of variables that go into postprandial sleep, including when they last ate, how well they slept the night before, time of day, and all of those many variables that are harder to control or expensive to control. And again, we were just fortunate that was something we could do in flies by having these hundreds or thousands of meal events that we could dissect and study from and then split further into meal components.
Yeah. You wish that someone would take this on, even though it's pretty challenging to do because of all of those variables that you mentioned in people.
Yeah, I would like to see some of the key experiments connected to meal processing or caloric processing or uptake from meals and how they affect both sleepiness and the actual change in physiology or metabolism.
So even if post-meal sleep is something that really can be observed in humans, and it does have some impact on metabolism, what then? For now, the answer is still, we don't know.
It's hard to say right now, just because we don't know quite enough yet about the biological relevance of the phenotype. It's just attractive right now because the phenotype, the behavior looks like it exists in multiple organisms. And so, we're still conjecturing, we're still speculating and hoping that means it's a relevant and important behavior for further study. But on the other hand, the human body, your body, tells you what it wants. And so if you get sleepy after eating, maybe you should listen to your body and take a nap. And we don't quite know what that does yet, but it might be important.
And this research lives on not just in the form of their continued work on post-meal sleep, but in this simple setup they designed that's been recreated by other labs to study their own questions about individual fly behavior. That's included works on the effect of social isolation on sleep, brain circuits controlling circadian rhythms, and age-related changes in metabolism.
Most of the stuff we do, especially for this project, are fairly inexpensive. It used to be 30 years ago when you made custom behavioral chambers, you'd have to have a machine shop make them for you out of plastic or acrylic, and they'd cut them for you and assemble them for you, and it'd be thousands of dollars. Now they have 3D printers that cost, $100 or $200. You can 3D print and test all sorts of custom chambers until you get the one that works best. The cameras can range in price, but we found that we didn't need high resolution cameras to do what we wanted. And so I think we were using Xbox cameras in our first version of the experiments that were, $50 or $100 each. So I really did enjoy this project and developing those methods, and that everything we done was pretty inexpensive, and that other labs, a few other labs picked up. So that's been really great to see when you develop a method and a few others, use it and are happy with it.
Thanks so much for listening. You may have noticed the large gap between my first episode and this second one, but PhD life got the best of me. I can see the light at the end of the tunnel though, as I'll be defending in September, and I've got plenty more to come before that. As a small gift to thank you for your patience, I'll be putting out a bonus episode with my interview with Dr. Christina May. You might remember her from the beginning of the episode.
This is what I want you to tell the world. Flies are cute.
We actually talked for almost an hour, and she had lots more interesting things to say about flies in general and her own research, so keep an eye out for that. You can find the entire transcript and a link to Dr. Jah's paper on our website, frivolousScience.substack.com. I'll also be uploading some videos of Dr. Jah's 3D printed fly array that was used in the study. Frivolous Science is made by me, Houda Khaled, with music by Blue Dot Sessions. Thank you to Josh Wilkerson for his fantastic voice acting, to all the fly scientists I talked to, including Dr. Kevin Nunez, Dr. Christina May, and of course, an extra special thanks to our guest, Dr. William Jah. I don't know how we got onto the subject, but he started complaining to me about his friend who never finishes his iced coffee because the bottom gets too watery.
They suck from the straw, they run away, they come back, they suck from the straw again.
Huh, couldn't be me. Well, I'm Houda Khaled, and this was Frivolous Science.